Alrighty, hello everyone. Welcome to tonight's lecture. My name is Megan and I'm going to be talking about glycolysis and the Krebs cycle tonight. So it's a biochem lecture. So this is our consent for audio video recording. This was for if people were going to join for tonight's live lecture, but it didn't seem like anyone was hopping on. So I'm just going to record this and upload this to YouTube for you to watch on your own time. So here I just have linked our GroupMe, our Instagram, our YouTube link, and the schedule. So we have 2 lectures a week, which are both actually going to be switched to recorded lectures that will be released at 8:30 p.m. Eastern on Sunday and Monday evenings. All right, so this is our question-based style course. So we're going to go through, I have 8 questions here that have some topic review following, and I have 2 review questions at the end to kind of go over some of the stuff that we'll be talking about in today's lecture. All right, so our first question here, which of the following lists the correct order of steps by which glucose is oxidized? So you can take a minute here, pause the video, and just take 1 to 2 minutes to answer the question. All right, now I'm going to assume that you've answered the question by now. So the answer here is A. So we go from glycolysis to the pyruvate dehydrogenase complex into the Krebs cycle and eventually end with electron transport chain. So I'm going to start doing a very broad overview of all of these, and we're going to dive into the specific steps and enzymes involved in both glycolysis and the Krebs cycle a little bit later in tonight's lecture. So for glycolysis, we start with 1 molecule of glucose and we end with 2 molecules of pyruvate, and the net energy product from this is 2 ATP and 2 NADH. And I say net because in the beginning, first 5 steps, we use up 2 molecules of ATP and we actually produce 4. So the net gain here is just the 2 ATP. Something important to note here is that you end with 2 molecules of the pyruvate. And moving forward, when we talk about the cycle, so in the pyruvate dehydrogenase complex, which is next, it just talks about 1 molecule of pyruvate going in. So it's really important to pay attention to whether a question is asking about how many things are produced per molecule of glucose, or if it's like per molecule of pyruvate, because obviously if it's per molecule of glucose, you'll have to double everything for the later steps like PDC and Krebs cycle. All right, so the pyruvate dehydrogenase complex, this one we're not going to go too in depth today because this is not super high yield on the MCAT, but it's really just important to know where it is in terms of glucose metabolism and the purpose that it serves. So basically here we have that pyruvate that was the end product of glycolysis. So that goes through, it's a complex of 3 enzymes, which we'll go over a little bit later. So the end product is acetyl coenzyme A, which is what is fed into the Krebs cycle. So we're basically just converting this pyruvate to something that can be used by the Krebs cycle to generate more energy products. So the PDC produces 1 molecule of NADH for each molecule of pyruvate. So again, here, just thinking about looking at question stems. So if you were asked how many molecules of NADH are produced in 1 round of PDC, it would be 1. But if it's asking how many molecules of NADH are produced from 1 molecule of glucose going through PDC, then it would be 2. So just pay attention to things like that. So next we have the Krebs cycle. I have a couple different figures of this that we will be looking at today. So this one is just a little bit more surface level. It doesn't go quite as in depth as we will go later today. So basically the acetyl coenzyme A comes into the citric acid cycle from the PDC, and that combines with oxaloacetate to form citrate. So the oxaloacetate basically is recycled throughout, so that's not something that needs to be brought in. The acetyl coenzyme A is the only thing that is being brought in from that glucose that we initially started with at the beginning of glycolysis. So for each round of the citric acid cycle, also you'll probably hear me rotate between calling it Krebs and citric acid cycle because it's the same exact thing, so it's just good to be comfortable hearing it as either way. So in each round of the citric acid cycle, we produce 1 molecule of GTP, 1 molecule of FADH2, and 3 molecules of NADH per cycle. So again, just to really drive this home, if this were asking about per molecule of glucose, that would be 2 GTP, 2 FADH2, and 6 NADH. So the electron transport chain, we're not going to talk about this too much today because this is actually covered in next week's lecture. So yeah, I'm just going to go over it pretty briefly today. So this, the electron transport chain uses energy from NADH and FADH2 that we've generated in some of these previous steps in order to pump protons against their concentration gradient between the mitochondrial matrix and the intermembrane space. So if you picture a mitochondria, there's that outer membrane and then there's all those like squiggly inside parts. So basically there's that inner membrane and there's a space between the outer membrane, and that's the intermembrane space. So we're pumping these protons against their gradient, which requires some form of energy. And that is so that they can flow out through this complex 5 at the end here, which is a source of energy, something moving with its gradient, and that allows us to produce ATP. Again, we'll talk about this way more in depth next week. I just wanted to go over it now just because it's really relevant. So rather go over it too many times than not enough. All right, so question number 2 about glycolysis. So question reads, glycolysis is specific to which domain? So I'm going to give you time here, pause the video, and take 1 to 2 minutes to answer the question. All right, so assuming that you have answered the question by now, I'm going to reveal the answer, which is D, all of the above. So I'm just going to go over a little bit about why that is and some helpful things to remember. So glycolysis it's universal across all domains. So all cells from all domains have the enzymes that are involved in this pathway. So we're going to go over all of those enzymes a little bit later, but all cells have them and are capable of doing this. So it is important to note here that oxygen is not a requirement in this step. So it's not an aerobic process. So it can occur in either conditions. And I've just put an equation up here that shows the net start and end products of glycolysis. So you start with 1 molecule of glucose and you have ADP, inorganic phosphate, and NAD+. And the result of that is 2 pyruvate, 2 ATP, 2 NADH, 2 molecules of water, and 2 hydrogens. So here I have the figure that we're going to be breaking down step by step a little bit later. So this is just good to familiarize yourself with. I know it looks like a lot right now, but we're going to really look at each step individually and make it a little bit more manageable. And I would definitely encourage you to use the slides from today's lecture and create your own sort of figure that just goes over, you know, how many carbons are in each molecule, what the enzyme is, which steps ATP is produced in, and things like that. All right, so question 3, this is just about cellular respiration in general. So the question reads, glycolysis occurs in which part of the cell? So take a minute here and pause the lecture. 1 to 2 minutes to answer the question. All right, assuming that you have now answered this question, I'm going to reveal our answer, which is C, the cytoplasm. So it's actually, this question kind of ties in well with the previous question. So glycolysis occurs in the cytoplasm, which is a good way for you to remember that it's universal to all domains because it's not dependent on any membrane enclosed organelles. So it can occur just the same in prokaryotes as it can in eukaryotes. It doesn't occur like in the nucleus or in the mitochondria or anything like that. It's just out in the cytoplasm. So yeah, that's pretty much all that we've covered on here. There is one difference between prokaryotes and eukaryotes regarding glycolysis, and that's in how much ATP they produce. So since the prokaryotes do not need to transport anything into the mitochondria, because eukaryotes actually carry out the PDC, Krebs, and the electron transport chain all within the mitochondria. So they use some energy to get those products in the mitochondria. And since prokaryotes don't need to do that, they actually end up creating 2 more ATP equivalents than eukaryotes. So I'll have all of these numbers with how many ATP equivalents, how many NADH, FADH2, and all that stuff is produced at the end of today's lecture as a little summary. So we're just going to go through where all these things occur. So pyruvate dehydrogenase complex in eukaryotes occurs within the mitochondrial matrix. So pyruvate enters the mitochondria through the MPC, which is the mitochondrial pyruvate carrier. Pretty nice and self-explanatory name. And so the reason that this is within the mitochondria is just because it allows the acetyl coenzyme A to easily enter the citric acid cycle, which is also taking place within the mitochondria. So for prokaryotes, the PDC is just located out in the cytosol because it's not really dependent on a membrane or anything. And yeah, something else to know is just that the PDC is one of the largest multi-enzyme complexes found in both prokaryotes and eukaryotes. So this exists as a complex of 3 enzymes. It's not just 3 loose enzymes, which just saves energy in passing products from active site to active site. So the Krebs cycle in eukaryotes occurs within the mitochondrial matrix, and in prokaryotes, it just occurs in the cytoplasm as expected. So yeah, here I just have a little picture that has a couple of these steps. So it just shows how this is for eukaryotes, obviously. So this showing how the Krebs cycle and electron transport are occurring within the mitochondria. So, okay, the electron transport chain, this one does depend on a membrane. So there's a difference with the eukaryotes and prokaryotes here. So for eukaryotes, this is the intermembrane space of the mitochondria. So as you can see here on the left, this outside is not outside the cell, but it's outside of that inner membrane. So that's in the intermembrane space. And that's where we're going to create our hydrogen concentration gradient that we'll be using. So yeah, and then on the left, you see that this is the mitochondrial inner membrane. That should say be a little bit more specific. And then inside is the mitochondrial matrix. So for prokaryotes, they just use their cell membrane to create this gradient and to pump their protons. So you can see here that the outside, in this case, will be the exterior of the cell, and then the cell membrane, and then the interior of the cell. So very similar, but just a little bit, just slightly different location here. Again, this will be covered in more depth next week, but I just wanted to go over it now while we're talking about where all of these steps are taking place. All right, so question 4, glycolysis, the preparatory phase. Which of the following enzymes catalyzes the first step of glycolysis? Take a minute here and pause the video and unpause the video once you've answered the question. All right, so I'm going to assume you've answered the question now and unpause or reveal the answer. So the answer here is hexokinase. So now we're going to take some time and go through all of the enzymes, and you'll see where all of these other enzymes come in. And just looking at this question too, one answer that you should be able to get rid of right away is this carboxylic acid, just because that is not an enzyme. So that's just narrowing down your choices wherever you can is a great strategy to have on the MCAT. You won't know every answer, but you should at least try to always use the process of elimination to throw out answers that you know just can't be correct. All right, so broad overview again. So steps 1 through 5 are the preparatory phase, and that is where we are using some ATP to get ready to produce more ATP and NADH. So that's why steps 6 through 10 are considered the payoff phase. So here we just have this same figure again, and now we're going to go through steps 1 through 5, which make up the preparatory phase. All right, so step 1 is where we start with glucose. That's our starting material. So it enters glycolysis when it is phosphorylated by hexokinase, which results in glucose 6-phosphate. So I have here glucose with an arrow to G6P. So at least for me, and a lot of texts do this as well, talking about things like glucose 6-phosphate as G6P, and it just makes it a lot easier to manage, especially when you're making your own figure for this and including all those enzymes and things like that. So it's important to note that this step is irreversible, and this costs 1 molecule of ATP. So step 2, so we have our G6P, which is then converted to F6P or fructose 6-phosphate by phosphoglucose isomerase. So that's good to know the enzyme, what we start with and what we end with. So again, I simplified it here. I have our G6P is converted to F6P. So step 3, we have our F6P, which is converted to F16BP or fructose 1,6-bisphosphate by phosphofructokinase 1. So as you can see in all these steps, I have our start, our end, and our enzyme all bolded, just so those really stand out. So this step is also irreversible and also uses 1 molecule of ATP. So this is where in our preparatory phase, we're spending or using up 2 molecules of ATP, and those are both in the 2 irreversible steps. That could help you remember where we're using that ATP. And they're both phosphorylations as well. So steps 4 and 5, some texts will combine these and say that glycolysis is 9 steps. But here we just have them separated out to make it a little bit more clear. So here that we have our fructose 1,6-bisphosphate, which is cleaved with the enzyme fructose 1,6-bisphosphate aldolase. So that'll make us 2 3-carbon sugars. So before this, we had our 6-carbon sugar. So when this is cleaved, it creates G3P and DHAP. So basically this additional step of step 5 is when DHAP is converted into GAP. So G3P and GAP are synonymous, and I meant to change that G3P, but just know that it's the same thing. So the DHAP is converted into GAP with triose phosphate isomerase. So basically moving forward, we just have 2 molecules of GAP that we're working with. So this step, yeah, is just F16-bisphosphate to our 2 molecules of GAP or G3P. All right, now we have our question number 5 regarding the payoff phase of glycolysis. Which of the following steps of glycolysis is irreversible? So take a minute here, pause the video, and unpause it once you've answered the question. All right, Assuming you've answered the question, I'll reveal the answer now. The answer here is the step where phosphoenol pyruvate is converted into pyruvate. So just looking at this, what I tried to do when I was studying for the MCAT was force myself to recall whenever possible. So just that forced recall and making yourself like kind of come up with questions where there aren't questions really helps solidify the information, especially when this information is new to you. It just makes it a little bit more familiar. So from earlier in the lecture, we talked about how pyruvate is the end result of glycolysis. So even if you weren't sure which step was irreversible, while you're paused and looking at this question, I would even use that as an opportunity to recall like, oh, like pyruvate, I recognize that. That's the last step. So just kind of forcing yourself to bring that information up whenever you can. And same with this glyceraldehyde 3-phosphate. Yeah. So that's our gap. I guess we didn't talk about the next step, but anyways, just trying to recall information whenever you can really, really helps solidify everything. All right, so now we're going to go over the steps 6 through 10 of glycolysis, which is the payoff phase. So step 6 is when we have our glyceraldehyde 3-phosphate, so GAP or G3P, and that's where this is oxidized to form 1,3-bisphosphoglycerate, so 1,3-BPG, and the enzyme here is G3P dehydrogenase. So at this step, we produce 2 molecules of NADH and 2 hydrogen. So this is important because this is one step where we're producing one of our energy products. So really, really important to remember which steps we're producing energy at. So here we have, G3P to 1,3-BPG. So step 7 is where we have our 1,3-BPG, and we have enzyme phosphoglycerate kinase, which transfers the phosphate from the 1 position of the 1,3-bisphosphoglycerate to a molecule of ADP. So this results in ATP and 3-phosphoglycerate. So a good way to remember this step, if you're just looking at the materials, is that you have 1,3-BP. So the BP means bisphosphate. So you have 2 phosphates. So you're transferring one of those phosphates to an ADP to make an ATP, so adenosine triphosphate. And then you're missing that phosphate from the 1,3-BPG. So you just have 1 phosphate left at the 3 position. So it's 3-phosphoglycerate. Not sure if that helps or makes things more complicated, but it's just being able to picture like the movement of that phosphate can help you remember that at least this is a step where we're producing ATP because we're losing a phosphate. So that phosphate has to go somewhere. So step 8, we have enzyme phosphoglycerate mutase, which converts 3-phosphoglycerate to 2-phosphoglycerate. So this is literally just moving the phosphate from the 3 position to the 2 position, which I think this enzyme actually kind of tells you what it's doing. It's just mutase. It's just mutating it. It's moving it. It's not really doing any super significant change here. So then again, we have 3-PG to 2-PG. So step 9, we have our enzyme enolase, which dehydrates 2-phosphoglycerate to form phosphoenol pyruvate. So phosphoenol pyruvate, we can write as PEP. So we have our 2-PG going to PEP. So also in these diagrams, something that I haven't yet mentioned that I probably should is that we have, I guess the quality of this is not great, but it's showing us our delta G. So basically when the delta G is positive, that's an unfavorable reaction. And that is why those steps that all have the positive delta G are not irreversible, because it actually would be favorable to move backwards, because a negative delta G is favorable conditions. So anyways, so pyruvate kinase, so step 10, pyruvate kinase is the enzyme that catalyzes the PEP into pyruvate. So we see we're at the end now. We've reached our pyruvate. That was our final goal. We made it. So this just transfers the phosphoryl group to ADP to form another molecule of ATP. And this is our irreversible step, because we see we have a pretty negative delta G there. So moving in this direction is favorable, so it's not going to want to move backwards. So just over the last 10 or so slides, when we went through all these steps of glycolysis, I would definitely go back through and use these simplified, like 2PG to PEP, and make your own figure, and maybe write the enzyme like on the arrow or something, and just draw out where ATP is produced, kind of like this. So I did a fairly more simplified version. So I have where ATP is consumed, where ATP is produced, and then the only thing I would really encourage you to add to this would be putting the enzymes involved on those arrows. All right, so question 6, we're going to talk about the pyruvate dehydrogenase complex. What is produced by the pyruvate dehydrogenase complex from one molecule of glucose? So this is what I was emphasizing before, to pay attention to if we're asking about one cycle or one round of PDC, or if we're asking about per molecule of glucose. So take a second here and pause the video, give yourself a minute to answer the question. Alrighty, assuming you've answered the question now, I'll show our answer, which is 2 NADH and 2 molecules of acetyl coenzyme A. So D is kind of a trap answer. So that would be just if we had one molecule of pyruvate going in, then we would only have 1 NADH and 1 acetyl coenzyme A. So again, we've talked about most of this already, but I'm still just going to go over it one more time. So it's composed of 3 enzymes that exist as a complex, and those 3 enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. We're not going to really go into the mechanisms of these, just because it's not very high yield on the MCAT, but glycolysis and the Krebs cycle are, so I really want to put our focus on that today. But it's just important to know that there are 3 enzymes in a complex, be familiar with the name of those enzymes, and that they exist in the mitochondrial matrix for eukaryotic cells. So yeah, they produce 1 NADH per molecule of pyruvate and 1 acetyl coenzyme A per molecule of pyruvate. So this is just shuttling that acetyl coenzyme A into the Krebs cycle. And here I have this same figure that we looked at earlier. So basically we have our starting pyruvate. We have our coenzyme A that comes in, our NAD that turns into NADH. And then we're left with our acetyl coenzyme A and carbon dioxide. All right, it's time to get into the Krebs cycle. Krebs citric acid or TCA cycle, whatever you prefer to call it. So which of the following enzymes does not result in a change in the number of carbons present in the molecules of the Krebs cycle? This is a tricky question. So basically we're looking for a step where the number of carbons present is conserved. So take a minute here, pause the video, and resume once you've answered the question. All right, so I'm going to show our answer here. which is B-aconitase. And we're going to now go through all of the steps, number of carbons, and enzymes present in the Krebs cycle. So here we have our overview figure, which I'm going to be using to break down all 8 steps. So basically just know that this can be called the TCA, Krebs, or citric acid cycle, and TCA stands for tricarboxylic acid. So here we're always starting with the acetyl coenzyme A that we generated in the pyruvate dehydrogenase complex, which is combined with oxaloacetate that we are recycling throughout the Krebs cycle. So what we're producing here is 3 molecules of NADH, 1 molecule of GTP, and 1 molecule of FADH2. And we are going to break this down into 3 stages to sort of make it seem a little bit more manageable. So stage 1 is where we are forming our citrate. So stage 1 is just step 1. It's basically this is where we're combining our oxaloacetate that we have recycled throughout the Krebs cycle with the acetyl coenzyme A from the PDC. And here we release that coenzyme A and we form citrate, which is a 6-carbon molecule. And here the enzyme is citrate synthase. So stage 2 is steps 2 through 4. So this is where we are oxidizing the citrate to release carbon dioxide and produce NADH. So we have 2 rounds of this oxidative decarboxylation. So we'll be forming 2 molecules of CO2 and 2 molecules of NADH. So basically it's 2 consecutive steps of essentially the same thing happening in a row. So step 2, citrate is just converted into its isomer, isocitrate, with the enzyme aconitase. So this is the answer to our question from this section. So we did not change the number of carbons, which is evident by the name of isocitrate. So if it's an isomer, you're not going to have a change in the molecular formula for that molecule. So step 3, we have our isocitrate is oxidized by isocitrate dehydrogenase, which forms alpha ketoglutarate. So here you can see, since we're releasing carbon dioxide, we have lost 1 carbon. So now our alpha ketoglutarate is a 5-carbon molecule. So then we're going to basically do this again with alpha ketoglutarate dehydrogenase, which will then release that CO2 and produce another molecule of NADH. And then our coenzyme A is added back into the mix here. But succinyl-CoA is our resulting molecule, and that is a 4-carbon molecule. So these 2 steps are really important to know because we are both changing the number of carbons in the molecule and producing 2 molecules of NADH. So stage 3, this is steps 5 through 8, and this is where we are regenerating oxaloacetate so it can continue going through the cycle. And we're also producing decent amount of energy products here with 1 GTP, 1 FADH2, and 1 NADH. So step 5 here, we have succinyl-CoA, which is converted into succinate with the enzyme succinyl-CoA synthetase. So as you can see so far in the Krebs cycle, a lot of the enzymes actually have their starting material or product in their name, which helps you remember where they go in terms of the cycle. So in this step, we're producing 1 molecule of GTP. Step 6, we have succinate is converted into fumarate using the enzyme succinate dehydrogenase. And here we're producing 1 molecule of FADH. It's also important to note that in both these steps, we're not changing the number of carbons at all. We still have 4 carbons present, and we will for the rest of the cycle. So in step 7, fumarate is converted to malate by the addition of a hydroxyl group. So we can see our H2O coming in here, and the enzyme here is fumarase. In step 8, our malate is converted back into oxaloacetate with the enzyme malate dehydrogenase. This is another step where we're producing an energy product, and we have one molecule of NADH. So I know that was a lot, but again, I would encourage you to do the same thing, as I said, for glycolysis, and make your own figure of the Krebs cycle using all of these steps, noting the number of carbons present in each molecule, the enzyme used, where energy, where we are getting our energy products, and of course, the names of all the molecules going throughout. All right, question 8. So this one is about the citric acid cycle again. How many ATP equivalents are produced in the Krebs cycle from one molecule of glucose? So again, pay attention to the question asking about one molecule of glucose and not one round of the citric acid cycle. So pause the video here and take a minute or two to answer this question. All right, so our answer here is C. So I'm going to go through how to calculate ATP equivalents from NADH, FADH2, and GTP, since, as we saw previously, there was no actual ATP produced in the Krebs cycle, but the MCAT loves to test on ATP equivalents. So basically, this is just the formula on how to convert these other energy products into ATP. So NADH has enough energy to produce 2.5 molecules of ATP. So however many molecules of NADH you have, you can multiply that by 2.5, and that's how you get ATP equivalents. FADH2 similarly has enough energy to produce 1.5 molecules of ATP. So however many molecules of FADH2 you have, you can just multiply that by 1.5 to get your ATP equivalents for that. And then GTP actually has a 1-to-1 ratio with how much energy it can, or how much energy it uses to produce ATP. So your however many molecules of GTP you have is equal to the number of ATP equivalents. So here, I would definitely encourage you to commit this chart to memory. So this shows here the actual molecules that are formed in glycolysis, the pyruvate dehydrogenase complex, and the Krebs cycle. And then over to the right, it shows the ATP equivalents. So you might be looking at this and seeing how we have our 2 NADH in glycolysis that only translates to 3 ATP, where you would think it would be 5 if you're multiplying it by 2.5. But this is what we touched on earlier, and how eukaryotes need to spend some energy to get all of these things into the mitochondria. So that is why in this case, only 3 are produced, whereas for prokaryotes, there would be 5 in this step. But in general, when not talking about glycolysis on the MCAT, 1 NADH equals 2.5 ATP. So that's the standard. This is an exception, but you should still commit this chart to memory. So this chart here is for eukaryotes. For prokaryotes, the only difference would be that 5 ATP from glycolysis, and then our total at the end would be 32 ATP rather than 30. All right, so now I have some review questions that are going to go over some of the stuff that we talked about in today's lecture to sort of help you practice recalling that information. Which of the following steps of the Krebs cycle depicts a change in the number of carbons? So again, here I would encourage you to try using process of elimination. If there are some that you know don't change the number of carbons, just cross those out and then use your best judgment to reach an answer. All right, I'm going to assume that you've answered the question now, so I will show our answer, which is B, isocitrate to alpha ketoglutarate. So if you remember, this was step 3. Yes, this was step 3. So this is one where we're releasing CO2 and producing one molecule of NADH. So isocitrate, that was where we still had our 6-carbon molecule, and we're going down to a 5-carbon molecule with alpha ketoglutarate. And one more review style question here. Which of the following involves the enzyme hexokinase? So hexokinase was actually asked about in our first question, I think our first question today, or one of them. Anyways, so yeah, take a minute here, pause the video, and give yourself a minute or two to go through all these answers, and see if you can recall which enzyme is used for each of these steps. That would be really great practice, using the multiple choice answers to force yourself to recall some of this information. All right, so I'm going to show our answer, which is A. So this is our first step of glycolysis. That's where we use the enzyme hexokinase. So I know I've said this a couple times during today's lecture, but again, I would encourage you to use these slides to create your own figure of both glycolysis and the Krebs cycle, making sure to include the number of carbons in each molecule, the enzymes involved, and at which steps energy products are produced. And just be familiar with the few subtle differences between cellular metabolism in prokaryotes and eukaryotes. So this was for our live lecture, which is not happening tonight. So next week on Sunday, the 16th, we have a Psych-Soc lecture that will be released on YouTube at 8:30 p.m. And that's about behavior therapy and psychotherapy. And then on Monday, the 17th, we'll have another BIOCHEM lecture, which will be our second metabolism lecture, talking about the electron transport chain and lipid metabolism. And I believe that will also be released on YouTube at 8:30 p.m. on Monday. So thank you all for watching, and happy studying.